Hey friends, welcome back to this channel, and this is another video on mechanism of action. We are dealing with the endocrine system. We have covered quite a bit of endocrine system, and we are now continuing with the mechanism of action. So in the previous video, we have covered the overview on the mechanism of action. We have discussed about the receptors and different locations of the hormone receptors, where we discussed that there are basically two locations where the hormone receptors are present. One is the nuclear receptor, another one is the membrane receptors, right? And then we discussed about those hormones which can bind with the nuclear receptor. We discussed about two different types of hormones. One is the steroid hormone, another one is the thyroid hormone. So we discussed that how they can bind with the nuclear receptors and how their things done. So today we'll be discussing about those hormones which can use these secondary messengers for their mechanism of action. Now, what is secondary messengers? We'll discuss these secondary messengers later when we will start our cell signaling system. But for now, you have to remember that secondary messengers are small molecules, means their molecular weight is very, very small. So they are the small molecules which are associated with the cell signaling system. And some examples of secondary messengers are like DAG, diacylglycerol. Then we have another secondary messengers like IP3. We have calcium. We'll discuss this in today's video. So we are discussing today hormones that uses the secondary messengers for their mechanism of action. And what are the secondary messengers? First thing is adenylate cyclase, cyclic AMP secondary messenger system. This is very important. I think most of you are quite acquainted with the term of G protein or G protein coupled receptor, GPCR. If you don't know all this term, don't worry, we'll get into those. Then we have the phospholipase C, calcium secondary messenger system. Third one is the tyrosine kinase secondary messenger system. And next we'll discuss epinephrine, how this epinephrine uses two secondary messenger systems together. So first thing we are discussing is adenylate cyclase, cyclic AMP or CAMP secondary messenger system. So let's see. So our body, in our cells, we have a protein that is called G proteins. Now in these G proteins, there are three subunits: alpha subunit, beta subunit, and gamma subunit. You have to remember that when this alpha, beta, and gamma are together, that is called trimeric G protein, because there are three subunits, right? Alpha, beta, and gamma. So that is called the trimeric G proteins. When these G proteins are associated together, this alpha, beta, gamma, all three are associated together, and they are forming this trimeric G protein. They are actually in inactive form. And where are they present? They are present in the cytosolic part of a receptor protein, and this receptor protein is actually a receptor for what? A hormone, right? We are discussing about hormones. So we have a receptor in the cell plasma membrane, and we have this trimeric G protein attached at the cytosolic phase of the plasma membrane. When hormone comes and bind with this receptor proteins, there are some changes. And what are the changes? Changes are this alpha subunit gets detached from this trimeric G protein, okay? And when this alpha subunit is detached, this alpha subunit becomes active, okay? And then this active alpha subunit can bind with another protein that is called adenylate cyclase. You know, this adenylate cyclase is nothing but an enzyme which can convert ATP into CAMP. ATP into CAMP. What is ATP? Adenosine triphosphate. What is CAMP? This is cyclic adenosine monophosphate. So obviously this is monophosphate, means there is one phosphate, and ATP, this contain three phosphate. So rest of these two phosphates are coming as PPI, okay? So pyrophosphate or PPI. Now this CAMP is acting as a secondary messenger over here. What is the role of this CAMP secondary messenger? See, it will get attached to the protein kinase, okay? Now this protein kinase now is remaining in the inactive form, and in this protein kinase there are actually two subunits. One we can see over here, this is the regulatory subunit, and another one is the functional subunit, okay? So CAMP will come and bind with this regulatory subunit, and it will detach this regulatory subunit from this protein kinase. First, let's see how this protein kinase remain. So whenever this protein kinase is attached with the regulatory subunit, it is in inactive form. What CMP does is, CMP comes and binds with this regulatory subunit and detach this regulatory subunit from this protein kinase. So this is the regulatory subunit and CMP complex. So this is now detached from this inactive protein kinase. As soon as this regulatory subunit is detached from this protein kinase, this inactive protein kinase becomes an active protein kinase and it can now do its function. So there are phosphorylation of proteins, and this phosphorylation of proteins, because this is a protein kinase is always associated with some phosphorylation. So since there is a phosphorylation of proteins, there will be activation of some specific enzymes or inactivation of the specific enzymes. So you can say that is how this adenylate cyclase cyclic AMP secondary messenger system is acting, and these hormones are actually having its effect on the protein kinase. Let's say this hormone will active this protein kinase. How this hormone is activating this protein kinase? Through this CAMP system, through using this CAMP secondary messenger system. So that is one thing. Okay, now the second thing is phospholipase C calcium secondary messenger system. Let's see the overall or the basic structures are same. We have a receptor, we have a hormone, and hormone receptor complex are bound together. So we have a trimeric G protein, and now the G alpha subunit is released and activated. So when this G alpha subunit is released and activated, it will come and bind with the phospholipase C. See the difference between this adenylate cyclase CAMP secondary messenger system and this phospholipase C calcium messenger system is the protein where the alpha subunit is bound. Okay, the active alpha subunit is bound. Let's say when we are discussing about the cyclic AMP secondary messenger system, this active alpha subunit binds with the adenylate cyclase. Remember, for them this adenylate cyclase CAMP system, the alpha subunit comes and binds with the adenylate cyclase, which converts ATP into CAMP. But in case of phospholipase C calcium system, this alpha subunit, active alpha subunit, comes and binds with the phospholipase C. Now what is the role of phospholipase C? This phospholipase C will help to produce IP3. What is IP3? That is ionocetyl triphosphate. That is IP3. Okay, so this is ionocetyl. This is basically an ionocetyl compound where there are 3 phosphate attached. So IP3, ionocetyl triphosphate. Now what is the role of this ionocetyl triphosphate? This ionocetyl triphosphate will come and enter into the endoplasmic reticulum, or we can say this is sarcoplasmic reticulum. Okay, and whenever there is a insert of AP3, whenever IP3 comes and interact with this endoplasmic reticulum or enter into the IP3, it will cause the efflux of the calcium channel. So we know that endoplasmic reticulum is actually the the bag where the calcium ions are stored, and IP3, what it causes, it kinds of open the channels for calcium efflux. So calcium will come out of this endoplasmic reticulum into the cytoplasm and will have their effect. And what happens if the calcium is increased in the cell cytoplasm? The cell will contract, right? Like in our heart system, in our cardiac muscle cells, there in our smooth muscle cells, everywhere in our skeletal muscle system, everywhere, whenever calcium is high, there is a contraction of the cell. So that's how the hormone will impact the cell, or hormone will have its effect via this calcium system, phospholipase C. This is the phospholipase C calcium system. Okay, so this is the second type of secondary messenger system that hormones use. Now next is another very interesting and intuitive thing, that is tyrosine kinase secondary messenger system. Now see, there is a difference between tyrosine kinase and protein kinase. In this protein kinase is kind of a general term, okay, which can this is an enzyme which can phosphorylate some other specific type of enzymes, but or specific proteins. But for tyrosine kinase system, tyrosine kinase are that kind of kinase enzymes or kinase protein which have a tyrosine site where it can get phosphorylated. Okay, so this is the tyrosine kinase secondary system. We are discussing about the insulin system or insulin regular mechanism of action of insulin, because this insulin is the classic examples where the tyrosine kinase secondary messenger system is used. Now I hope you remember the dimerization thing that we discussed in our previous video. Please go and check it out. This is that dimerization part is very important. Okay, so this is the insulin receptor. These two are insulin receptor and they are now separated, and so two molecules of insulin come and bind with the alpha subunit. So remember that this insulin receptor contain two subunits or two chains. One is the alpha chain, another one is the beta chain. The alpha chain form the binding site, insulin binding site, and the beta chain is kind of regulatory site, okay, or the functional site. So insulin comes and bind with both this alpha chain, and as a result there is a dimerization. So both these insulin and its receptor chain comes and get dimerized together, and so there is a phosphorylation of the receptor. See, that's why I said that tyrosine kinase is a bit different, because there is a phosphorylation of the receptor. Previously when we discussed about the adenylate cyclase CMP system, we discussed that there is protein kinase is active and protein kinase is causing the phosphorylation of some other proteins. But in this case of tyrosine kinase, at first, before some other proteins are getting phosphorylated, the receptor itself is getting phosphorylated, and that phosphorylation is occurring at a tyrosine residue. Okay, so there is a tyrosine residue, both these tyrosine amino acids are getting phosphorylated, so that's why the name tyrosine kinase secondary messenger system. So there is a phosphorylation of the receptor, and now the tyrosine kinase is activated. So for this phosphorylation, again 1 ATP has to be converted into ADP. For this phosphorylation, 1 ATP is getting converted into ADP, and both these receptors are getting phosphorylated at the tyrosine residues. Okay, remember this. Next, as the phosphorylation is done, phosphorylation of the tyrosine residue are done, now this tyrosine kinase protein or the insulin receptor is now activated, and it is now phosphorylating some other protein, other specific protein, to its phosphorylated form, and for that also 1 ATP is getting converted into ADP. So that is a cascading effect. That means maybe this particular phosphorylated form of a protein will go and phosphorylate some other protein. So that is a cascade mechanism, and due to that cascade mechanism, glucose uptake and anabolic reactions will occur. We all know that whenever there is a huge amount of blood glucose level, insulin spikes and it causes the glucose uptake and anabolic reactions. So all those anabolic reactions and glucose uptake happens because there is a because this insulin level increases, insulin binds with this receptor, the receptor gets dimerized, and after that their tyrosine residues get phosphorylated, they gets activated, and those phosphorylated tyrosine residues with and the activated insulin receptor will cause the phosphorylation of other signaling molecules that will initiate a cascade effect, and then the glucose uptake and all the anabolic reactions will happen. So I hope that makes sense. Let's discuss about epinephrine. I hope you remember epinephrine from our previous video where we discussed about how the hormones can interact. We discussed about the antagonistic effect, synergistic effect, and permissive effect. So in synergistic effect, I hope you remember about the epinephrine. I discussed that epinephrine individually can increase the cardiac rate. Norepinephrine can also individually increase the cardiac rate, but when epinephrine and norepinephrine both are present, the increase of cardiac rate will be more than both of their individual effects. Okay, so let's now discuss about this epinephrine and how epinephrine uses two secondary messenger system. So this is the liver cell where the epinephrine actually works, and we are discussing about the alpha adrenergic effect of epinephrine and the beta adrenergic effect of epinephrine. See here, first the beta adrenergic effect of epinephrine is using adenylate cyclase CAMP system. That means the ATP is getting converted into CAMP via adenylate cyclase, and that CAMP is activating the protein kinase, okay, and that protein kinase is causing the conversion of glycogen to glucose 1 phosphate. Okay, so this is the first secondary messenger system that we discussed, that is the adenylate cyclase cyclic AMP secondary messenger system, and that is being used by what? That is being used by beta adrenergic effect of epinephrine. Now what about the alpha adrenergic effect of epinephrine? They are basically using the phospholipase C calcium secondary system. So by using that system, the calcium level is increasing, and whenever there is a increase in calcium level, another protein that is called calmodulin is a protein which is dependent on calcium. Whenever calcium is increased in the cell cytoplasm, it will go and bind with the calmodulin, and it will increase or it will activate the calmodulin. So we often say calcium calmodulin complex. Okay, so this calmodulin will again activate the protein kinase. So you can see in both these cases, no matter this epinephrine uses this adenylate cyclase system or the calcium system, phospholipase C calcium system, the ultimate effect is the activation of the protein kinase, and that protein kinase will convert this glycogen into glucose 1 phosphate, and then the glucose 1 phosphate will be converted into glucose 6 phosphate. And we know that in the neoglucogenesis we have discussed that glucose 6 phosphate 5 phosphatase can be converted into free glucose, and then free glucose from the liver cell, it can go to the blood, and through blood actually it can go to all the cells which require the glucose. Okay, so this is a very interesting and intriguing information, that epinephrine can use both this phospholipase C and calcium secondary messenger system, as well as the adenylate cyclase cyclic AMP secondary messenger system. But remember, alpha adrenergic effect is for calcium and beta adrenergic effect is for adenylate cyclase. Hope that makes sense. Hope you get an overall clear picture of how these hormone which uses the secondary messengers can have their own role in our body. And okay, so this is it for today's video. In our next video we'll discuss about different endocrine glands. If this content was helpful, please make sure to subscribe, like, share and comment. See you in the next video. Bye.